In the case of transcription of eukaryotes, there are a bit complexities, two additional complexities when compared to the transcription in prokaryotes. The first complexity is that there are three RNA polymerases for the transcription of different types of RNAs. RNA polymerase 1 transcribes the rRNA, that is ribosomal RNA, of which the components are 28S, 18S and 5.8S. We know rRNA or ribosomal RNA are the building units of ribosomes. Ribosomes are made up of rRNA and proteins. So these are the big units of the ribosomes which we had learnt in class 11. And RNA polymerase 2, it transcribes a heterogeneous nuclear RNA, that means hnRNA. RNA polymerase 2, it brings about the transcription of the hnRNA, that is heterogeneous nuclear RNA. It is a precursor of the mRNA, that is before processing. It is the precursor of the processed or the final mRNA. And RNA polymerase 3, it brings about the transcription of tRNA, that is transfer RNA, the 5S rRNA, that is part of the ribosomal RNA, and in addition to it, small nuclear RNAs. So these are the three different types of RNAs: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. In addition to it, the next complexity is that the primary transcript, that is the hnRNA, heterogeneous nuclear RNA, or the nascent RNA or the new RNA which has just been transcribed, it contains exons and introns because the eukaryotic structural gene is monocystronic, which consists of the coding sequences and the intervening sequences. So exons and introns, as we had learnt earlier, isn't it? We learnt that in the final mRNA or the mature RNA, only exons are present, that is the coding sequences, whereas the introns are totally absent. So in the case of eukaryotes, the primary transcripts consist of exons and introns and are non-functional. Such an mRNA is non-functional. That's primary transcript is non-functional. It cannot undergo translation. So before undergoing translation, it has to be processed and all the introns has to be removed. So these are the two complexities when compared to the prokaryotic transcription. Three RNA polymerases are there which codes for the different types of RNAs, and the primary transcript is non-functional because it is containing introns along with exons. So the processing of the primary transcript, the steps involved are the same: initiation, elongation and termination. After termination, what is formed is the hnRNA. We can see the exons and the introns, and this is the 5' end and this is a 3' end. So this nascent RNA or the hnRNA which has been formed has to undergo processing. So three steps are there in processing. First is splicing. Splicing is the process by which all the introns are removed and all the exons are joined. So exons are joined together and all the introns are removed by the spliceosome. By the spliceosome, all the introns are removed and exons are joined. That is a process called as splicing. And a cap has to be added at the 5' end. This is a 5' end. And a cap has to be added. What is this cap? It is methyl guanosine triphosphate. It is a cap, methyl guanosine triphosphate. We can see this guanosine with 3 phosphates with a methyl group attached to it, and it is attached to the 5' end of the nascent RNA. And the last processing step is a tailing process. Here we can see the tail. The tail is made up of adenylate residues. Adenylate residues, about 200 to 300 adenylate residues are added to the 3' end of the hnRNA in a base independent fashion. So that is the 3 steps which is done for processing: splicing, capping and tailing. And after these 3 processes, the hnRNA is converted into a mature mRNA, that is a messenger RNA, which is ready for translation. In the case of eukaryotes, we know they have a true nuclei in which the nuclear material is surrounded by a nuclear membrane. So in the case of eukaryotes, unlike the prokaryotes, there cannot be coupling of transcription and translation. The transcript which is formed over here has to be brought out of the nucleus into the cytoplasm, where the machinery for protein synthesis is present. That is, ribosomes are present in the cytoplasm. So it has to be transported from the nucleus to the cytoplasm. Only then translation can take place. So dear children, in this module, we learned about the process of transcription which takes place in prokaryotes and eukaryotes. Thank you.